So we've learned quite a bit about how proteins fold, but we've done so under assumptions that are based on a homogeneous solution of dilute protein that's folding in an aqueous solution, for example, in an aqueous buffer in our test tube in the lab. So now we're going to consider folding in the environment of the cell, so how the protein deals with the cellular environment. We often think that the cytosol is this big wide open space for the molecules to float around, but in fact it's actually jam-packed with molecules, particularly proteins and RNA. So this image represents a simulation of the crowded cytosolic environment in a bacteria. So this crowded environment poses some unique challenges to proteins as they fold, and that's the subject of this video. So the challenge here is that the protein has to fold onto itself to achieve its own tertiary structure. And that's in contrast to it might very well interact with other proteins or other molecules in the process of trying to fold, and that would inhibit its folding ability. Here's our model of the protein folding funnel, where the protein can achieve its native fold, the energy minimum. And we've seen that this is true in dilute solutions, for example, in the Enfinson experiment. But in the cytosol, there are many other proteins that are in close proximity to the nascently folding proteins, the proteins that are just synthesized by the ribosomes. So if the protein chain actually interacts with other proteins that are also nascent folding proteins, they could adopt other conformations that have lower energy, in particular some multi-protein aggregates. And this potential problem is illustrated here by extending the conformational space available to the protein in the process of searching its native fold. So we've added a whole other side to this graph showing the potential for aggregates. The same energetic forces that dictate native protein folding dominate also in aggregates, namely the benefit of burying hydrophobic residues through the hydrophobic effect. So this suggests that in the cytosol, there's a risk that proteins aggregate rather than adopt their native fold, and then they couldn't perform their biological functions. But aggregates are even more nefarious than that. For example, here's an electron micrograph of aggregates of a peptide called amyloid beta. And these aggregates are one of the pathogenic features of Alzheimer's disease. So you can see that aggregates can be really deleterious to cellular functions. So how do cells actually prevent these aggregates from forming? So cells can prevent the aggregation using proteins called chaperones, and these chaperone proteins will actually aid protein folding. But let's not assume that each protein has its own chaperone to bend it into its native state. We know that the native state is thermodynamically favorable. That's the thermodynamic hypothesis. But what chaperones can do is prevent the newly forming proteins from aggregating with other proteins. So we'll start with one of those chaperones, HSP70. It plays an important role in folding of the proteins as they are being translated, right as they're coming off the ribosomes. And mRNA here is translated by a ribosome. And so you can see the polypeptide coming out. And the polypeptide that is synthesized is at risk of forming some aggregates. And that's especially true with large proteins because they have complicated folds often. So for example, if the fold of a protein would require interactions between distant elements in the sequence in the primary structure, for example, between the N-terminus and the C-terminus, the N-terminus is translated first, and it may form some aggregates with other proteins before the C-terminus can be translated. And that's where HSP70 comes in. So when HSP70 is there, it can bind to the nascent protein before it's fully translated and before it aggregates. So the protein will then be more likely to reach its proper fold. But how would it do that? So the HSP70 interacts preferentially with hydrophobic residues. It will transiently mask these hydrophobic residues and prevent aggregation. But the key word here is transient, because HSP70 also needs to release the substrate to allow it to try to fold. If it was stably bound, it would effectively be just another kind of aggregate instead of the aggregates that it should prevent. So how does HSP70 interact only transiently with its substrates? It uses cycles of ATP hydrolysis and exchange of ADP for ATP. ATP hydrolysis causes a massive conformational change that allows substrate binding to HSP70. The substrate binding, in turn, will promote the exchange of ADP for a new molecule of ATP. And now HSP70 changes its conformation back to a state that is not permissive for substrate binding. That will cause the release of the substrate. The released protein then has another chance to fold on its own and bury its hydrophobic surfaces. This schematic here illustrates that HSP70 can exist in either a substrate-bound or a substrate-free state. And the structures of these two states have actually been determined. So let's take a closer look at the structures to see how ATP hydrolysis and exchange affect substrate affinity. Here is HSP70 in its ATP-bound substrate-free state. It has an ATPase domain that's shown in blue and a substrate binding domain that's shown in red with a yellow lid domain. Now with a transparent ATPase domain surface, you can see that the ATP is actually quite deeply buried at the center of the molecule, at the center of this ATPase domain. And upon ATP hydrolysis, the yellow lid actually moves into close contact with the rest of the substrate binding domain, the orange part of the protein here. And that forms a substrate binding pocket that sequesters some of the substrate's hydrophobic surfaces. So this is the part where HSP70 is sequestering the nascent protein. Now if we return to the ATP bound structure, notice how the exchange of ADP for ATP moves the lid domain away and abolishes this pocket. It is remarkable how the state of the nucleotide affects very distal part of the structure. With the ATP and ADP bound structures superimposed here, you can see just how massive that conformational change is between the ADP bound state, which is the solid surface here, and the ATP bound state, which is the transparent surface. The movement of the lid domain is what creates a binding pocket that is crucial for that transient affinity of HSP70 for its substrates. So let's unpack the name. HSP70 means heat shock protein 70. There's actually a whole class of heat shock proteins, and most of them are chaperones. But where does the name heat shock protein come from? It's because the expression of these heat shock proteins, or HSP proteins, increase with exposure to high temperature. It's usually a transient exposure to high temperature, so a heat shock. So here you can see it in a Western blot experiment. So the cells were heat shocked at time 0, and then the expression of HSP70 was monitored over 8 hours. And you can see that especially at 2 and 4 hours, there's more HSP70, there's a darker band, than there was at time 0. That's in contrast to actin, which is the control here, whose expression is about constant throughout the experiment. But it's important to see that HSP70 is also present at pretty high levels in unstressed cells at time 0 here. And that shows that it's important in folding, in helping proteins fold, even without a heat stress. So why is HSP70 expression increased so much with heat stress? So heat can cause proteins to denature. This plot shows the fraction of protein that's folded versus temperature for a representative protein within the cell. So as the temperature increases, there's more denatured proteins or fewer folded proteins. And that's dangerous because these denatured proteins could form aggregates through intermolecular hydrophobic interactions when the cells cool. So the cells will upregulate chaperone expression to prevent this aggregation and allow proteins to refold intramolecularly so that they regain their native tertiary structure.
Are chaperone proteins important in other situations?
Yes, actually they are, but let's just recap first. So we've already seen that under basal conditions, they're important to maintain the homeostasis of the cellular protein content, and that under heat stress, that's a condition that actually led to their discovery. They're very important under heat stress. But they're also important in response to other stresses, and you'll see increased chaperone expression in these situations too. One example is in secretory cells, when they're very active, when they're generating a lot of secreted proteins. That can cause some protein folding stress. Translating so much of the secreted protein could lead to aggregates. So part of the stress response to this increase in secretory activity is to increase chaperone expression to assist with protein folding. So remember that HSP70 is not the only chaperone. Actually, mammalian genomes have on the order of 200 chaperone genes. One really interesting chaperone is called HSP60, or also called GROEL in bacteria, and it's shown here. It forms a huge multimeric cage structure, and this view here is a cutaway view that shows the inside of that cage structure. A whole protein can go inside this cage to fold into its native shape in a safe environment that's free from hydrophobic interactions with other proteins. Let's recall the thermodynamic hypothesis, and that's the hypothesis that the most thermodynamically stable state of a protein is this protein's native fold. And for most proteins that are folding in dilute solutions, this is true. In the crowded environment of the cell, chaperones assist folding in several ways. One of them is that they help proteins as they fold, overcoming the kinetic traps that are along the pathway. But even more important is that chaperones prevent the thermodynamically favorable aggregates from forming, and that's why many of the chaperone genes are actually essential genes. Here's a fun analogy to close out this video. So you can think of the crowded environment of the cell as the mosh pit at a rock concert filled with teenagers having a lot of fun. So the chaperones are just preventing the teenagers from getting too tangled up before they've grown into full mature adults.